All right, so welcome to the Protist lecture. This is what I'm calling the second set. And the reason why I split it was just to make these file sizes smaller so they're more manageable in Blackboard when you're downloading, etc. It's all covering the Protist, but there's just a lot of content here. So it's easier to kind of compartmentalize the lectures into different parts. First part, remember, that covered our intro to Protists, kind of their evolutionary history, characteristics, big general characteristics, and then also the supergroups excavata, and we started and did about half of the chromioalveolates. So what I want to do with the second set is continue the chromioalveolates and then start talking about the other supergroups that we need to look at with the Protists as well. So the first part, we left off with this review slide, and we were talking about chromioalveolata, and I said, all right, what's the key feature? And then we went down to the subcategory alveolata, again, features, and then we talked about these three groups here that all fit within alveolata: dinoflagellata, the apicomplexans, and the ciliates. So that's what we covered on the previous set. Well, what we're going to cover now is chromioalveolata, but we're going to go a different direction. We're going to go to the right, and we're going to talk about the straminopilla, or straminopiles. We'll get into these guys in just a second. But within straminopilla, or the straminopiles, there are going to be three groups as well. So what we want to do is look at the three groups that sit within straminopilla. We're going to have the brown algae. Oh, let me change font. Okay, so we got brown algae. We have the diatoms. And we have this weird, crazy group called the oomycetes. Okay, so those are the three categories within straminopilla that we're going to start discussing here. So they're chromioalveolata, so they have that feature, but to be straminopilla, they're going to have a different feature. Actually, let me do this so we color code, make a little more sense. Let's use that one. All right, so we're going this direction because there's a different feature. Once we're here, we go either here, here, or here, based on other characteristics and features that we'll talk about for each of these three groups. Okay, so we're just going now to the right-hand side of the review slide, tackling those three groups and looking at their diversity. So, all right, so let us begin with what makes a straminopilla, or the straminopiles. And the defining feature is that they have hairs on their flagella. That is our big feature here for this particular subcategory of the chromioalveolata. So they're still chromioalveolata, but then to come here, they need these little hairs on their flagella. So you can see it here, little picture, little micrograph, those little hairs sticking off of the flagella is the key feature of the straminopiles. Now within straminopiles, the first group we want to tackle will be the brown algae. These are generally called seaweeds, or sometimes you'll hear the term kelp. So brown algae is the largest and most complex algae out there. So these things can get up to 100, 150 meters long. I mean, they can be massive. If you go out to the Pacific Northwest, you get into the kelp forests within the Pacific Ocean, and these things literally make forests in the ocean. They anchor to the bottom, and I'll show you a structure in just a second, and then they can grow up hundreds and hundreds of meters to reach the surface. And they're so thick they look like trees literally in the ocean. Oh, these guys are going to be multicellular. So when we talked about protists, most protists are single-celled. The algae, the brown algae, they ignore that rule, multicellular. And the vast majority are going to be marine organisms. Okay, now I want to insert a blank here. Okay, so the way these brown algae work is they live in the marine, and the vast majority live in the marine environment. And what these guys will do is they anchor themselves. Let's make this the bottom of the ocean. And if you guys have been out to the Pacific, a lot of areas where you find these big algae, it's a rocky bottom, big kind of rocky substrate. It's not smooth, nice sandy beaches where we're going to find these algae. And what they're going to do is they're going to anchor themselves to those rocks with a structure known as a holdfast. It is not a root. It resembles a root system, but it's an anchor, and it literally will grab on and attach itself to the rocks. That allows it to hold place in the water so it doesn't get washed around and blown around with this particular type of brown algae. Then along the length of the algae, it's got these little, we're not calling them leaves because that's appropriate for plants, but just little projections of their body. The more of these that they have, the more surface area for photosynthesis. And if we zoom into one of these, let me make the big, this is a zoom in view. What we often find are these little bladders. They look like little pimples. They're air bladders. They're filled with gas. And this allows the algae to float. So each of these little extensions here will have several of these things embedded within them. So the algae is able to float. So what's going to happen here is the algae will float based on where the tide is at. So if the tide is low, then the algae will float like this along the surface of the water. Because those little bladders keep them at the surface of the water. Now, as a tide goes up, then the algae upright or orientate themselves upwards because of those little bladders. So it's a great way for these guys to constantly be close to the surface of the water. The biggest reason here is when we look at the brown algae, they contain this brown pigment called fucoxanthin. Fucoxanthin is not the best pigment for photosynthesis. It's brown. It absorbs some wavelengths of light, but just not the best, which is why these things need to be as close to the surface as possible. If you're deeper in the water, light penetration doesn't work. The wavelengths of light don't get all the way down to the bottom in deep water. So as light hits the surface, it starts to lose wavelengths, and the deeper you go, the more wavelengths you lose. So you wouldn't be very effective photosynthesizer way down here, 100, 200 feet below the surface, because the light just can't reach you. So you use a brown pigment to try to absorb the light, and you always are floating up and down near the surface of the ocean. For those of you who are fans of sea turtles, little baby sea turtles love brown algae. It's supposed to be a sea turtle. Give him a little beak. There you go. Okay, use your imagination, turtle. And the reason why they love these brown algae is that there is a type called sargassum that will create these little floating islands out off of the Atlantic coast. So go to East Coast United States, Florida, North Carolina, etc., and go out into the ocean. There is an area known as the Sargasso Sea, and it is made up of brown algae. Sargassum is the type of brown algae. The little turtles swim from where they're born on the coastline all the way out into the ocean, and they go into the Sargasso Sea. They hang out, they have protection until they can get a little bit bigger and then try to survive swimming around the ocean and eventually return to those beaches to lay their eggs, and then their little babies swim back out to Sargasso Sea. So take a look at it on NOAA's website, Oceanography Maps, and you'll see an area in the middle of the Atlantic. Again, it's called the Sargasso Sea. It's a region of the Atlantic where the ocean currents cause the sargassum to just kind of stay in this big, I mean, we're talking 100-mile radius, if not bigger, area where this stuff collects. Those algae are not anchored. Those brown algae are just floating at the surface, but they're still using those little air bladders. All right, so to get an idea of what the anchor looks like, this is it. It's officially called a Holdfast. So let me put that term here. So that anchor structure So it anchors the brown algae to the substrate, whatever that substrate may be. And generally it's just rocks. So these are pictures I took when I was in Washington State years ago. We went out to the coast, and this is what the algae structure looks like. So not a true root, just an anchor. And then you have the blades. And then if I went further up, you'd see the floats. And that's going to be the general structure of the brown algae. When it comes to life cycle, they go through what's known as an alternation of generations. And what we will see is the sporophyte stage, where it has its full genetic material. It's going to be diploid, 2N. It's going to be this multicellular structure. It produces these little tubes called sporangia. That's where it's going to produce the spores, equivalent to eggs and sperm. Those get released, they go undergo meiosis, and they can either be male or female gametophytes, gametes, which are going to be haploid, half the genetic material. The male-female need to combine. Now there's a challenge of doing this in the water, but they combine, they fertilize, and then they come back and form the sporophyte, which begins the next generation. So they alternate, sporophyte, haploid generation here. So diploid, haploid, when they're going through their life cycle. Okay, so those are the brown algae. Let's take a look at the second group within the straminopilla, and these are going to be the diatoms. Now, diatoms are an incredibly important marine organism. So these guys are going to play a huge, huge role in marine ecosystems when we talk about the producer base. So I'm going to break it here, and then I'll start the second part, which will be about 15 more minutes, and we'll talk about the diatoms.